Yeah.
Okay.
Is that better? being streamed, I believe, on YouTube, if I'm not mistaken. I'm Trent Vorlicek I'm the representative of the Department of Chemistry, or Biochemistry, Chemistry, and Geology here at Minnesota State. And it is my honor to introduce today's speaker, Dr. Christian Abnet. He is I believe this is our 32nd iteration of the Ford Lectureship. So he is our 32nd Ford lecturer. Congratulations. Dr. Abnet comes to us today having gotten his undergraduate degree from the University of Oregon. He then went to the Midwest and got his Ph.D. in Environmental Toxicology from the University of Wisconsin in Madison. He then went back across the Mississippi and got his Master's in Public Health and Epidemiology from the University of Minnesota, Twin Cities. He is presently the Chief Officer for the Metabolic Epidemiology branch of the National Cancer Institute, affiliated with the National Institute of Health. To give you an idea of Dr. Abnet's academic bona fides, he has, I believe, 400 publications plus, and most impressively, those 400 publications have 40,000 citations. So that's a really impressive ratio, to say the least. A journal would be very proud to have that ratio of citations to publications. I think without further ado, I will simply introduce Dr. Abnet. If we could also, I want to mention that tonight at 7:30 p.m., we have a second talk, which is more of a general talk, that Dr. Abnet will be giving, because we like to extract as much as we can out of our Ford lecturers. Without further ado, Dr. Abnet.
All right. Thank you very much.
I don't think...
No, I don't need that. No. Okay. Thank you very much for the introduction. And so my field is cancer epidemiology. I knew I was going to be speaking with a mixed audience today, so I thought I would try to tell you about what cancer epidemiology is, and why we do it, and how it complements other forms of cancer research that you may be more familiar with. So this is my maternal grandfather. His name is William A. O'Brien, and he was the head of graduate medical education at the University of Minnesota, and he was the head of the Minnesota Cancer Society. And he might have had a big impact on me, except that he died in 1947, and I never met him. But something about him was always kind of rattling around in the back of my head, and I ended up following in his footsteps in some way. He liked to teach undergrads, and at the time it was required that all undergrads take a class called personal hygiene. And it was basically the kids had to learn how to take a shower, brush their teeth, and all sorts of things that you don't think of as a university activity. What he did was so amusing that somebody made a movie about it once, and the movie was so bad that it ended up on Mystery Science Theater, Body Care and Grooming, and you can watch it on YouTube. It's really funny. It basically says, if you don't take a shower, you're not going to get any dates when you're in college. So very good stuff. Okay, so as I mentioned, I went to the University of Oregon, and then Wisconsin, and then to Minnesota. And I was kind of wandering around trying to figure out what I wanted to do. I went to Oregon because I was interested in marine biology. They had a program that let you spend 1 year on the coast of Oregon, Jamaica, and Massachusetts to learn marine biology, and I did that. On the right is my favorite professor. He studied animal behavior. He always studied lobsters, and he says he never did an experiment he didn't eat. So he would work on the lobsters, and then he would take them home and cook them. But he was gaining so much weight, he was going to switch to tuna the last time I saw him. So after that, I wasn't ready for graduate school. I knew I wanted to go to graduate school, but I became a wildlife biologist for the U.S. Forest Service, and I spent a couple of years chasing owls, trying to figure out where their nests were. And it was a really fun job, but you had to be nocturnal, and I had to spend the whole week out in the woods with mice, because that's how you find their nests. And I knew that wasn't a long-term career option for me. So I went to Wisconsin, and then I got closer to chemistry. I studied this compound, which is 2,3,7,8 tetrachlorodibenzopyridioxin, which is a very, very potent developmental toxicant, and I studied how it would affect fish. So I wasn't studying cancer, I wasn't studying humans, and it was an interesting work. I did structure-activity relationships, I cloned the receptor, from both rainbow trout and zebrafish to try to understand how this chemical worked differently between fish and rodents. When I finished my PhD, I knew that it wasn't for me, and I thought that I'd really made a big mistake. I didn't want to be a bench scientist because I just didn't think I was that good at it. So I had to find something else to do, and I found a program at the National Cancer Institute that took people with doctorates and retrained them to do public health research. So the first thing they said is, go to a public health school and get a master's of public health. And so I went to the University of Minnesota because my grandfather had been there, and because I had a brand new baby, and my family was here because I'd grown up in Minnesota. My wife's from Mankato, and it was a lot easier if we came back here. So we did that. Then I went out to NCI, and I never left. I thought I'd be there for maybe 3 or 4 years as a postdoc, but I got a good job, and so I didn't leave. And now I'm the chief of this group called the Metabolic Epidemiology Branch. We use a semi-academic system, a tenure system for our investigators. I have 12 principal investigators in my group. Altogether, we're about 50 people, and we are responsible, as it says here, for defining the relationships between diet, energy balance, which means physical activity, hormones, tobacco, and cancer. So we use epidemiological methods to understand what causes and prevents cancer. Okay. The National Institutes of Health is principally a training organization. We get a tremendous amount of funding. The vast majority of that funding goes out as grants. We keep a little bit of it for ourselves to do research. And when we do research, we do a lot of training. So I have a group of about 50. These are the 41 fellows that went through my branch in the last 4 years. You can see that it's quite a diverse group of people. I'm in a field that's completely dominated by women now. It's not really clear why, but you can see that it's almost all women who've been coming through our training program. We're a very quantitative group. Our principal activity is biostatistics, and it's a changing face of our science. So far, 14 of these people have gone out to start as assistant professors at various institutions. Okay, so when you think of cancer research, what do you think it is? Most people, their first thought is this: a physician by a bedside with a cancer patient, and they're trying to keep them alive. And they're trying to define new ways of treating cancer. And so here we have a little guy who's probably got a childhood cancer like leukemia, and they're trying to do something new to cure that cancer and keep them alive. This is what we call clinical research. It's very important, and it's probably where most of the money in cancer research goes. Second is bench science. So we have a lot of basic scientists who are funded to use cells and other model systems to try to understand how cancer forms, how can you interrupt it, and that aspect of it. And so there's a lot of biochemistry here, and that's sort of the basic science. So we have basic and clinical science, but there's a third branch of cancer research, and that's what I do, which is epidemiology. Epidemiology is defined as the branch of medicine that deals with the incidence, distribution, and possible control of diseases and other factors relating to health. So our goal is to first count diseases to see what the burden of disease is in a population, and that's called descriptive epidemiology, and then we use analytic epidemiology to try to inform what's going on with those patterns. John Snow is considered the father of the field. He was a physician in London during a cholera outbreak. And he went out and he asked all these households if they had a cholera case, and then he asked them where they got their water. And he figured out that all the people getting cholera were getting their water from a pump called the Broad Street Pump, because this is before water was piped into homes. And he thought he could do something about it, so he took the handle off the pump so that the pump didn't function, and the cholera epidemic abated. And he said, oh, I've done something. So first he counted, and then he did something. And so that's important. Our other sort of founding father is Dr. Osler, and he said medicine will become a science when doctors learn to count, which we like to, because we like to poke fun at the MDs. And the idea is that a physician practicing medicine is not doing science. They're using science to treat patients, but they're not actually doing anything scientific by treating the patients. They have to do something experimental or observational to make it into a science. Okay, so the burden of cancer dramatically varies by organ site and geographic location. So if you haven't really thought about this, you might think that cancer is just something that's bad luck. It happens to people, and there's sort of this even distribution of cancer all over the world. Well, that isn't what happens. Cancer is driven by specific exposures in most instances. There are some cancers that we really don't understand at all why people get them, like glioblastoma in the brain. It's pretty much a complete mystery why people get glioblastoma. But other cancers, we can sort of figure it out, and we want to describe the burden. So why would we describe the burden? So here's an example. This is the incidence rate of lung cancer by county in the United States. For reasons I don't know, Minnesota and Kansas had suppressed their data from this database, so I don't have the data for Minnesota, but you can see that there's a very uneven distribution. Now, this isn't the count of cancer cases, it's the incidence of cancer, that's a rate. It's the number of cases per 1,000 people, per 100,000 people actually, but the same idea. And you can see that Kentucky has a really high rates of lung cancer. That dark red means there's high rates. And if you look at Utah, for example, there's almost no lung cancer. And so this has three important reasons for knowing that. One is we know the health of our people, and we know what kind of burden of disease they're facing. So that's important. And why does that matter? Well, if you're running a hospital and you're in Kentucky, you better have people who can treat lung cancer. If you're in Utah, maybe it's not so important. You could have one or two centers that focus on lung cancer because there's just not that many patients. The other thing that's important, not exactly at the state level, is for industry. If you need to know where the burden of the disease is, because if you want to work on something to cure a disease, if you work on lung cancer, you're going to have a lot of patients. If you work on small bowel cancer, which is very rare, you're not going to have a lot of patients. And so they have to make business decisions based on the burden of cancer. The burden of cancer changes not just with geography, but it changes a lot over time. So this is the distribution of cancer death in men since 1930. So in 1930, Connecticut started counting cancer deaths, and by about World War II, all the states had joined in, and all states are responsible for counting the cancer deaths in their country and in their state, and they get basic information on people. And you can see that it's changed a lot. In 1930, stomach cancer was by far the leading cause of cancer death in the world, or in the United States. And by this point, in the 2000s, it's rare. It's a very rare cancer. We have a pretty good idea why that is. But if you look at lung cancer, in 1930, it was almost non-existent. Almost nobody got lung cancer. But by 1990, it had gone 50 times higher. And we know why that is. It's because cigarette smoking. In 1930, machine-manufactured cigarettes were just becoming available, and people were starting to smoke a lot, and the more they smoked, the more lung cancer we get. In 1964, the Surgeon General said, smoking causes lung cancer, and that changed the pattern. We were at the peak of about 60% of men were smoking in 1960, and that's fallen dramatically. Now in some states we only have 10%. In Kentucky, we still have 40% of the adults are smoking. And so it took about 30 or 40 years before the cancer rates start to come down after the behavior changes, because you have this built-in exposure history that you have to process through the population. So all this stuff is very interesting. You can see in other things, there was a spike in prostate cancer. That's because of PSA testing. There was a lot of guys who had prostate cancer and they were dying without knowing it. And when you start testing for it, it shows up. And their deaths might get attributed to it, even though it maybe wasn't what killed them. And colorectal cancer, actually the incidence was going up and it started going down because of colorectal cancer screening. So when you first start screening, you find a lot of cases, but if you keep screening, you can actually reduce the incidence. If you have a colonoscopy and you have your polyps pulled out, you'll never get the cancer. So it can really change things. So epidemiology is responsible for describing these patterns, and that's descriptive epi. But we try to do more than that. John Snow did an intervention where he took the pump handle off, but we also can do something called analytic epidemiology, which is more than just observing it. We simulate experiments because we can't do experiments necessarily. Okay, so how do you learn what causes cancer? You can ask a cancer patient why they got cancer, and you might get an answer that makes sense, but it'll be a mixture of what they know, superstition, and things that they either feel bad about or feel offended about. Some people think their cancer has occurred because they did something wrong, and they'll be dwelling on that. Some people think it's something that was done to them from a workplace exposure, and that's possible. But for the most part, it's not really an accurate way to figure things out. It can be good for the patients to talk about it, but it isn't what we want to use to decide if something's a cause of cancer. Okay, but what do I mean by cause? Okay, so for something to be a cause, there has to be a biological mechanism. Sometimes we observe an association, but we can't figure out a mechanism, and maybe there isn't one, but there must be if it's really a true cause of the cancer. So usually with enough work, we can figure it out. At first, people couldn't figure out how viruses caused cancer, couldn't figure out how radiation caused cancer, but with biochemical and other types of work, you can figure this out. There has to be a predictable dose-response relationship between exposure and outcome. If you have a group of people with a low level of exposure and another group with high level, you should have corresponding differences in the cancer rates. That's another indication that you have a cause. But what you don't need is that everybody who's exposed gets cancer. Cigarettes cause lung cancer. But if you smoke one cigarette, you're not going to wake up tomorrow with lung cancer, or maybe ever. That's just not how it works. It's a probabilistic statement, and we have to accept that that is the way cancer and many other diseases work. You don't get a one-to-one correspondence. If you talk about tobacco and lung cancer, often you'll get somebody say, my grandma smoked for 50 years and she didn't get lung cancer, so I don't think tobacco causes lung cancer. And you say, well, how did she die? Well, she had a heart attack. And you say, well, cigarettes kill more people through heart attacks than through cancer, but anyway, that just doesn't mean it's not a cause. That's an anecdote, and she was fortunate, but that doesn't refute the idea that cigarettes cause cancer. And as a thought experiment, what if I say, last year, 43,000 Americans died in automobile accidents. It would be totally illogical to say, my grandmother was in an automobile accident and she didn't die, so I don't believe that car accidents killed anybody, right? It doesn't make any sense. But these are things that we have to kind of help people think through. And it's really important because sometimes this turns into big money when we have regulations and lawsuits and all these things. So we have to be careful about when we say something causes cancer. So going back, how do we figure out what causes cancer? Okay, so if you didn't know where to start, you might start with cells. So this is more like the basic science research. If you have a hypothesis about a chemical that it's dangerous, you might start by culturing cells, putting some of the chemical on the cells, and seeing if it damages the DNA or causes them to replicate quickly or something, and you have some sort of alteration, basic scientific research. It's very helpful. But it doesn't by itself lead you to draw a conclusion about a cause of cancer. It's informative, and it's really important for that mechanism part, but it's not enough by itself. So what else can you do? Rats. So many rats and mice have been sacrificed to help us understand cancer. You can take genetically identical rats, house them in identical conditions, and the only thing that varies is their exposure to this chemical, and you can get that dose response I was talking about. And if you can do that, that's another important piece of the pie. But if you expose a rat and they don't get cancer, does that mean it's safe? No, because rats are imperfect models for humans, and they're helpful, but they're not necessarily great. For example, we know that heavy alcohol consumption causes esophageal cancer. Of the squamous variety, which is my main area of study. And rats have keratinized esophagus, and you can keep a rat drunk for 2 years and it will never get a esophageal tumor. It just doesn't happen. There might be some strain that does, but it's just not going to work. And that doesn't repudiate the notion that we know that heavy alcohol consumption can cause cancer. So now, if we can't use just lab or just animals, we have to use people, and that's where epidemiology comes in. We want to use free-living people who are going about their lives and try to use them to understand what's going on more generally. There's lots of nice volunteers who are happy to help us with cancer research studies. It's not hard to find them. And so what do? We take this group, we divide them in half, and we tell half of them you have to smoke cigarettes for 40 years, and we're going to see what happens, right? Now, that would be unethical. We cannot do experiments on people. Of that variety. You can do experiments on people, short-term experiments to look at exposures. You can ethically try to randomize people to cancer-preventing exposure, doing something to prevent cancer, like a chemoprevention trial, where we give them a medication we think will prevent cancer. That's okay. But we can't do causation through experiments in humans. So does that mean we're stuck? No, there's something we can do, and this is what I call analytic epidemiology. And one way to go about that is to do what we call a case control study. So we set these up at a hospital, and you collect everybody who comes into the hospital who has a specific type of cancer, and you invite them to participate. People generally are willing to participate in these studies as long as you don't catch them immediately after their diagnosis when they're distraught, but often they'll come around and participate, and you can ask them to participate, and then we get a reference group of healthy people, and we ask them all the same questions, or we collect blood from them, or urine, feces, saliva, all sorts of stuff, and we can compare the exposure rates between the two groups, and that can give us some insight into whether or not it likely causes cancer. If it was a lung cancer study, a lot more of the cases will have a history of smoking than the reference group, and then you can quantitatively estimate the risk of tobacco smoking. So that's good, but it's fraught with problems. When you ask a cancer patient how they live their life, it's very difficult. They'll either overestimate or underestimate the things, the way they live their life, because it's just hard to remember how you ate and how you did things. And there's some social desirability involved, and so it's not ideal. Finding a right comparison group is difficult. The people who volunteer to be in studies tend to be really healthy. What we really want is just the average random person off the street, and we used to do random digit dialing, just cold calling people to see if they would participate in studies, which was great. These days, nobody will answer the phone, and they won't do it, so you can't do that anymore. So it makes it difficult to find this comparison group. A better way to go about this is to do what we call a cohort study. Forget about the cases. Now we problem of people who are already ill. Instead, we just take a huge group of healthy people, we ask them the same questions, we measure the same exposures using biosamples, and we follow them, and we wait to see what happens. And we don't tell them what to do. We don't do anything special necessarily with this group. We just follow them. And so what that takes is a lot of time. Because if you have a big group of people, any individual cancer is a relatively rare outcome. Even if you had, for example, a whole group of people who smoke 2 packs of cigarettes a day, most of them aren't going to get lung cancer. Only about 10% of them will. And so you have to wait a long time, and you have to have a lot of people. And I mean a lot of people. So typically, if we want to do a cancer study, we'll have at least 30 to 50,000 people in our study, and we'll follow them for 5 to 10 years or more. In fact, we go a lot bigger than that sometimes, and we study them for decades. Now, you don't do that to do one disease. Usually you do lots of different diseases with a cohort study. This is a very effective way of doing an observational study, but it does take a tremendous amount of patience, and that's why we do a combination of different things. We have the bench work, we have animal work, we have case control studies, and we have cohort studies, and we have patterns analysis of descriptive epi, and we try to synthesize all that together to make decisions. Okay. So is epidemiology the weakest link? And the answer is yes, it is. It's the weakest link because we're not doing an experiment. I can't force those people to smoke cigarettes. I can't do a randomized study of actively exposing people. So yes, it is a problem. And a lot of times, if you dare to look at the comment section in a newspaper about a cancer study, a cancer epidemiology study, the first thing you'll read is correlation does not equal causation. Which is true, but it's not so profound because we all know that. And so it's like, okay, good, what are you going to do next? It's true, and some people seem to think correlation means it's not causal. I mean, no, because if you have a perfect causal pathway, there'll be very correlated exposure and disease because that's what's supposed to happen. And no, just seeing a correlation doesn't make us conclude that something causes cancer, he didn't just fall off the turnip truck. So let's think it through a little bit more deeply. Okay, and here's a good example of a correlation that's not causation. I like this one a lot. Ice cream sales peak in August, and so do shark attacks. And it's not because sharks like a cream-filled victim. It's because it's hot, and people like to eat ice cream and go swimming when it's hot, and so they're correlated. Yeah. So they're not, that's right. It's true, correlation does not equal causation, but you got to do more than that. Otherwise, you're basically just giving up and saying, we'll never know anything. Okay, so I'm going to give you now, switch over from sort of a general discussion of how we go about things to giving you a specific example from my work. So my specialty is the esophagus and the stomach. And these are 2 cancers that are not so common in the United States, but they're very common in other parts of the world. So I end up doing a lot of my research in other parts of the world where these diseases are common. When you look at the NCI budget, it's focused on diseases that are common in the United States. Breast cancer, colon cancer, prostate, and so forth, pancreas. Pancreas isn't super common, but almost everybody dies who gets pancreatic cancer. And so the ordering of the funding is very similar to the ordering of the disease in our population, which makes sense. But we do spend a little bit of money on rare diseases because sometimes rare diseases in unusual populations can tell us something that we wouldn't know otherwise. Okay. So the first symptom for esophageal cancer is dysphagia. So this on the left is a picture of a person with a big tumor in their esophagus. And the problem is that your esophagus is distensible. So when you swallow a big load of food, then your esophagus can open up to let that pass. And if you get a tumor in there, it's still going to be moving for a while. So at first, you can keep eating even though you have a big tumor growing in your esophagus. Eventually, it's going to get too big. Food's going to start getting stuck. And that's usually when you find out, and you go to the doctor and you find out you have esophageal cancer. Typically, people don't even live for a year with esophageal cancer because the tumor has gotten so big and it can go out, it can wrap around your aorta, it can cause all sorts of other problems. And so this is, it's not a certain death sentence, but it's pretty bad. And very few patients are treated with curative intent. Most of the time when you find people with esophageal tumor, we do palliative care on them because it's too late to try to cure them. On the right is a picture of a flat esophageal tumor that's tiny. You can't even see it as sticking out from the mucosa. We've stained the esophagus with iodine, which helps us target that. And I have a big aspect of my program is figuring out how to do early detection and intervention. But that's more like a clinical research aspect, not epidemiology. So this is a little bit old data, but this is the worldwide deaths due to cancer. 1.5 million people per year die of lung cancer. 85% of that is because of tobacco smoking. A little bit of it is because of radon. Some people it's secondhand smoke, some genetics, and some other types of exposures that are pretty uncommon. There's almost 1 million people per year die of stomach cancer. This mostly happens in destitute areas of the world. A lot of this is in China because China has high rates and tons of people. Colon and rectum is rapidly rising all over the world because it's correlated with a Western lifestyle of sitting too much and eating too much. And liver cancer is sort of a mixture. Liver cancer used to be primarily caused by hepatitis and aflatoxin. Once we knew what aflatoxin was in places like the United States, we have excellent agricultural systems that keep aflatoxin out of the food supply, so we don't have to worry about it. And we have hepatitis B vaccinations, which prevent hepatitis B infection. If you give it to the babies, you just have to wait 90 years for all the exposed people to process out of the population. But liver cancer is being controlled there, but liver cancer is also caused by obesity. So we have a new problem driving liver cancer. Okay, and then the esophagus. The esophagus, it kills about the same as number of people as breast cancer, but as I said, it's very rarely researched because it's rare in the United States. Okay, this is a map of the world. The high incidence areas for esophageal cancer are in China, the Middle East, East Africa, South Africa, and it used to be in Uruguay down in South America, plus the UK and France. The UK and France have a very different type of esophageal cancer problem. It's the problem that we have mostly in the United States. It's called esophageal adenocarcinoma. It's a glandular cancer. It just almost didn't exist until about 1980, and that's a very different disease process than the squamous cell carcinoma. So the normal esophagus is lined with squamous cells, and you can get a tumor that grows in those squamous cells. That's what most of this is, and that's what I study. So my particular story I'm going to tell you is about zinc, and this is a picture of a toy made out of zinc called the atomic disintegrator, and you'll see why I picked this for the title for this section because of some of the technology I used to study zinc. So when I was a postdoc, I was reading up on esophageal cancer. As I said, I didn't have any background in cancer, and I was reading a lot of papers, and I kept reading papers from this professor named Louise Fong. And she was at Ohio State and Thomas Jefferson, and she made her whole career studying esophageal cancer in rats, and her main interest was in zinc deficiency. And she had noticed somehow that zinc deficiency potentiates the effect of carcinogens. So in this experiment, she was using NMBA, which is N-nitrosomethylbenzylamine, which is a known carcinogen that targets the esophagus in rodents. And by giving graded doses of NMBA, you can develop tumors at graded levels in groups of rats. Zinc deficiency makes this worse, so rather than feeding them a well-balanced chow, she had special chow made that was zinc deficient, and she brought their zinc status way down, and that made them much more susceptible to the effects of the carcinogen. If you look at the tumor incidence on the right, in each row, there's first a zinc-deficient rat group, and then there's a zinc-sufficient rat group. And so zinc deficiency alone, she had 13% tumor incidence in the esophagus. And then if you look at the other two groups, again, the carcinogen gets dramatically potentiated by having zinc deficiency. This is a relatively small study. She did this many times. It's very predictable how this works. So when I read this, I thought, well, we know that NMBA is an experimental carcinogen. People aren't exposed to it normally in the environment, but there's a lot of related chemicals, and that could be acting on people. And I thought, well, we also know that there's a lot of parts of the world where people's diet is terrible, and maybe they have zinc deficiency, and maybe that's part of why we have these clusters. So there's one part of China where about 10% of all people die from esophageal cancer. It's this incredible cluster, and we thought, well, maybe, and they have a terrible diet, maybe they're zinc deficient, and maybe that's the problem. So I wanted to see if I could translate what was well studied in rats into humans and see if I could make some sense of what's going on. So the first thing that we could do is do a trial. We could take 30,000 people and randomize half of them to getting more zinc, and the other half we just leave alone. And then you do that for 5 years, and you probably could figure out whether or not zinc replenishment helps prevent cancer, and then you would indirectly infer that it was the zinc deficiency that made them at risk for cancer. And in fact, so that has been done on a large scale, but it's very difficult and very expensive, and you have to have a really advanced hypothesis before you can get the millions of dollars you need to do an experiment like that. In the United States, an experiment like that will cost between $80 and $100 million, and they're very rarely funded. The last one that was funded by NCI was for vitamin D, and it showed that vitamin D doesn't do anything to prevent cancer, which wasn't really too big of a surprise, but people are obsessed with vitamin D right now for some reason, and so it was worth funding it because we had to see what would happen. But I, at the time, was basically a brand new assistant professor equivalent, and they weren't going to give me millions of dollars to do this study of a rare cancer in the United States. I had to come up with something observational I could do. So I could, because my colleagues had been collecting material in China for decades at that point, and I could then use what was built up in our biobank to try to address the zinc question. And we had tissue, and we had food frequency questionnaires, which tell us about what people eat, and we had blood. And so could I do a study using this cohort of people where I knew what their lives were like at baseline, assess their zinc exposure, and then follow them? And I didn't have to start from scratch because all this already existed in our biobanks. And so I had to think about, how do I measure zinc exposure in a human? Okay, one thing to remember with nutritional exposures is that we don't have a normal dose-response curve. For an adverse exposure, you start with low exposure, no disease, high exposure, high disease, but that isn't the way nutrients work. Some nutrients like vitamin C, it gets better and better for you, and then you just pee it all out. If you take more vitamin C, maybe there'd be something adverse happens if you take a lot. But for something like a mineral, usually what happens is if you get too high a dose, you'll get intoxication. So this is an example for a mineral. You'll start off with low concentrations. You're in the adverse area, and the green represents whether you're going to have an adverse effect or a beneficial effect. And when you're below the deficiency level, you may have a deficiency-related disease. Like for vitamin C, you'll have scurvy. And if you don't get it up, the vitamin C intake up, you're going to be in trouble. And so you'll go up. At some point, you'll pass what we think is sufficiency. But then for some exposures, some nutritional exposures, you'll start to hurt you to get more, and you get to the point of intoxication. So for selenium, if you eat too much selenium, your nails crack, your hair falls out, and you can get really, really ill. And different parts of the world, people are going to be in different areas. So the Chinese farmers I'm studying, they have a bad diet because they mostly just eat corn, grain, and they have very little else in their diet. So they're deficient because of that, and they have very low distribution of a lot of nutrients. NHANES is a representative sample of Americans, and there you'll have a wide variety. You'll have a small number of people who are deficient or insufficient, and most people will be right in the middle. People who volunteer to be in studies in the United States are healthy, and they usually have good diets unless we really go out of our way. So they'll have a higher distribution. And then we have people who like taking vitamins because they think that those are good for them, and they'll have really high levels, and a few of them will take way too many vitamins, and they may be actually at a toxic level of intake. Okay, so just something to keep in mind. When good, we normally think of as better, that means a different thing in a Chinese farmer than it does in a US cohort participant. Okay. So if we want to estimate the zinc people eat, it's really helpful if it's inherent to the food. So some things like iron the amount of iron in a piece of meat is inherent to the meat. And you can see that these are studies. On the left, this is red meat, red and beef, and those have high levels of heme iron, and it's very predictable that cow flesh is going to have a predictable amount of heme in it. And so if you knew how much red meat somebody eats, you can estimate how much heme they got from that food. If you have, this is mostly pork, it's going to be lower. You know, the other white meat doesn't have as much heme in it, so you're going to get a bit less heme. And then if you have chicken, the chicken thighs have a fair bit of heme, but chicken breast has very little heme. And so if this was true throughout the food supply, if you know what people ate, you can estimate how many nutrients they took in. But zinc is not inherent like this. Okay, it doesn't work. And here's an example of something else that doesn't work, as I mentioned, selenium. If you grow wheat in North Dakota, there's a ton of selenium in the soil, and the wheat's going to be full of selenium, and the fodder from there can actually intoxicate the cows. Whereas if you go to eastern Washington, there's not so much selenium in the ground, and the wheat grown in eastern Washington will have low selenium, and that's important. Here in the United States, integrated agriculture, you don't know where your cremettes came from, but it's about, on average, it's sort of the same. If you're in a population that's subsistence farmers, this could be a real problem because they can only eat what they grow themselves. Okay, so it doesn't work very well for selenium, and it doesn't work for zinc. The other thing is that zinc has variable bioavailability. So if you have zinc-rich food like oysters, and you eat some oysters, you're going to get a huge dose of zinc, and that's good to a point. But this is showing you that the fraction of absorbed zinc goes down as phytate goes up. So if you're eating whole wheat, the phytate is going to block the zinc absorption. So even if you knew that somebody ate oysters all the time, if they also ate whole wheat bread all the time, it's going to block some of that zinc uptake. And so for all these reasons, zinc doesn't work for nutritional intake estimates the way you could with heme iron, vitamin C, some other things like that. So this doesn't work. So I had all this diet data, forget it. I can't use it for zinc. What about serum? We have big banks of serum from thousands and tens of thousands of people that we followed. I could just measure the zinc in their serum and away I go. No, because it's homeostatically maintained. You want your zinc in a narrow window, so if you have excess zinc, it gets put into your bones. And then if you have a zinc deficiency, that'll come back out of your bones, and it'll be a long time before you actually look deficient in the serum. So zinc, if there's homeostasis, it doesn't work very well to measure it. You can, if you eat a ton of iron, you'll generally have higher iron than somebody who doesn't, even though iron's also homeostatically maintained. But you're really taking out all the dynamic range has been crushed down to a homeostatically maintained range. Another thing we do for minerals is we measure it in toenails. So I have tens of thousands, hundreds of thousands of toenails that I've collected over the years, and you can measure stuff in toenails, and that is a good biomarker for zinc. But the farmers I study farm barefoot, and they also don't have enough water to bathe all the time, so their toes are covered with dirt. And when I measure their toenails, I'm measuring dirt. I'm not measuring a biological concentration. So, okay, forget that goes out the window. I can't do that. So what am I reduced to? I'm reduced to tissue. Now, Louise Fong had showed that what happens with zinc deficiency is that when the deficiency reaches the esophagus, your DNA repair enzymes start to fail. And that's why you get this problem, because the DNA repair enzymes are no longer working because there's not enough zinc to make these enzymes work. So working in the tissue would be perfect. And now I just needed to measure the tissue level of zinc. And I had one of my colleagues who does a lot of endoscopic biopsy collections, had a big collection of biopsies. He said, sure, they're about a square millimeter, and you can have 5 microns. So I have a pretty small piece of tissue, and I had to figure out how to measure the zinc in that. I can't grind it up and measure it because it's just too few molecules of zinc in this tiny piece of tissue to use atomic absorption or something like that. But there is a way to measure it, and that's with X-ray fluorescence. So X-ray fluorescence, you hit something with an X-ray, the elements glow, and then you measure the glow, and you can figure out the concentration. This is an X-ray fluorescence analyzer, and I think this may be used to do lead paint in walls. You hold it up to the wall, and you zap the wall. And that's cool, but I didn't have a wall. I had this tiny piece of tissue. So I talked to a physicist, and he said, you just need bigger X-ray machine. You just need enough power, and we can do this. And he said, I have one. It's outside of Chicago. It's called the Advanced Photon Source. Okay? So this is the instrument, and this is the parking lot. And this is a 1K ring, and here's a linear accelerator, and they get photons moving really fast. And then there's tangents, the X-rays are coming off, and all these notches are experimental HUDs. And a lot of this is done for crystallography. It's because you could get really intense X-rays. He said, oh, your problem, that's no big deal. So I applied. I got 4 days in an experimental HUD here, and I went in and we measured the zinc content of those little tiny pieces of tissue. So that was pretty cool. We only had 4 days, so I could only do, we estimated 200 measurements. We ended up doing about 130. I had 72 people that over time, over 15 years after their biopsy, didn't get esophageal cancer, and I had 60 that did. And there's some comparisons about who they were and what the tissue looked like. And then what I did is I measured the zinc concentration and I put them into groups. Q4 meant they had high levels of zinc in the tissue, and the 3 to Q1 would be the lowest quarter. And I compared their follow-up experience, and you can see that the people who had a lot of zinc in their esophageal tissue, only 10% of them got esophageal cancer over 15, 16 years. But the people who had low zinc, 35% of them got esophageal cancer. And now this is a very special subset. It's not a typical piece of the population because I picked people to participate in the study based on who got cancer and who didn't. And then we estimate the hazard ratio, and essentially having high zinc makes you 1/5 as likely to get esophageal cancer, or if you turn it around, having low zinc makes you 5 times more likely to get esophageal cancer. So I had read these cool experiments in rats. I wanted to translate it into humans. I couldn't do the experiment, so I did this observational study. I had to go through a lot of rigmarole to figure out how to measure the zinc, but I figured out how to do it, and this was very exciting. I published this in 2005, and then I kind of hit a wall. There aren't that many people out there who've collected tons of biopsy tissue from people who are at high risk for esophageal cancer. I couldn't repeat this, and to really believe epidemiology, we need to do it more than once. We need to do it repeatedly. And what happened is I basically haven't really followed up on this very much. A few other people have done something similar. People started doing it for colon cancer because there's a lot of colon tissue around, so that makes it easier to do. I ended up following other hypotheses. I'm a disease-focused researcher, so if I hit a wall with one thing and I think I've made some progress, I just move on to something else because there's always some other question I can answer. The other thing is that having a normal level of zinc in your diet, we already think that's good. Do I really have to kill myself to show that having a normal level of zinc is a good idea? Probably not. Did I do enough to convince the Chinese government that they should fortify the grain in this area with zinc? No. We have done enough to convince them to fortify the grain with selenium because we have a lot of other data that shows that selenium deficiency is a serious problem for esophageal cancer. The country of Finland had low selenium, and they were having a problem with heart disease, and so they started putting selenium in the fertilizer, and within 5 years, they had brought up the population level of selenium so that almost everybody's sufficient, and their heart disease rates went down. So what we are at now is we're trying to convince the Chinese government to mix selenium and zinc into the fertilizer in this part of the world, and then we would do what's called the demonstration project. We'd start on one end of the province by fortifying, and we'd move our way across the province and see if we could get the disease rates to change years later following that pattern. Because having normal nutritional levels, you don't really need to prove that. It's already generally a good idea, and other people have already figured out how much to put in the fertilizer so you don't intoxicate people by putting in the fertilizer. We thought about doing a salt intervention, putting it in the salt just the way we put iodine in the salt, but this coincided with the food adulteration scandal in China. Didn't go over well. So the hope is that we'll eventually do a demonstration project to show that this is effective. But other than that, I'm kind of stuck. Okay. So that's just one example of what the kind of work we do. I now have moved on to I do some nutrition. I do a lot of work on the human microbiome, which is all the bacteria and viruses that live in and on us. If you listen to my talk tonight, I'm going to talk more about that, the work I've done to try to understand why bad teeth puts you at risk for cancer. And I just want to finish up by saying, as I started, the NCI is a training organization. There's 4,000 postdoctoral fellows at NCI or at NIH right now. We have usually 20 at a time in my group. We have 1 or 2 people usually who aren't postdocs. We have people who come and do their Ph.D. research with us. We also have a lot of summer interns that come in for people who are undergraduates if they're interested in seeing what cancer research or other types of research is like. It's pretty competitive, and it's not competitive as in having the best CV. It's just so many people apply. It's kind of hard for people's applications to rise to the top. I do epidemiology. The vast majority of our internships are in laboratories because that's where most of the research is happening. But if somebody's interested, I'd encourage you. The DCEG training site is here. There's a general NCI and general NIH training sites. There's a lot of opportunities for people who want to get involved. I think that's what I was going to tell you today. Our division does all sorts of different types of epidemiology, and thank you for listening. Sure.
When you look at zinc in tissues, I'm just curious, like for example, the toenails or the esophageal cells, does that represent zinc within the last day? Does that represent zinc within the last month?
Yeah.
Lifetime?
Right. So in fact, esophageal zinc, we don't know because there's been so few studies studying something like that in tissue like that. For the toenails, we do have a good idea about the exposure window because it's been important enough that nutritionists have done those studies. Usually we use the big toenail because it's thicker, so you get more material. The end of your big toenail represents pretty much what you ate about 1 year before. And it integrates something like a few months worth of diet into a normal toenail trimming. And so that's there we know. In the esophagus, we're not really sure. This homeostatic process probably means that it's varying somehow in concert with the blood, but we're not really sure because it hasn't been worth figuring it out. Yeah.
If anybody else has any questions, we've got the microphones. If not, that's fine too.
Sure.
I'll just talk real loud.
Okay.
Is the province you're talking about in China, is that the Keshan province?
Well, there's Keshan disease. I'm working in it's Hunan province, and the town is called Linshan. But yeah, Keshan disease. That's where they get Kakisaki virus reactivation, and that's because of the selenium deficiency. Yeah. So the area where I work, the selenium is low, but it's not quite that low. So they don't get that cardiomyopathy problem. They're running at about they're just a little bit above, right around deficiency levels. So it's they, for the most part, they don't see these other problems that you might see with severe selenium deficiency. And so we think it's really an interaction with some other exposure. They make pickles in a really awful way, and we think that it's probably there are a lot of nitrosamines in the pickles as part of the problem, because they don't use vinegar. They use wild yeast to grow their pickles, and so all sorts of weird stuff grows in their pickles, and we think that's part of the problem.
So when you're choosing people for your cohort in the U.S., how do you choose them? Like, do you choose them by geographic location? And if they move, then do you remove them from the study? Or
Yeah, so it varies on what we're doing. So some cohorts are done by picking people in a specific occupation. I don't work on it, but we have it's called the Agricultural Health Study. We have licensed pesticide applicators in Iowa and North Carolina, and it's almost all men, but we also recruited their spouses. And so that's one way we built a cohort, and now we have a cohort of people who have much heavier than typical pesticide exposure. So that's one way we would do it. We've just started a new cohort where we want to collect 200,000 Americans. And we want people where we can get their medical records. And so what we did is we asked health maintenance organizations if they wanted to partner with us. Health Partners has agreed to be part of this project here in Minnesota, and also Sanford, the health system in South Dakota, is part of it. And so we're recruiting the people from within those health systems, because that way when we get their approval, we can access their medical records. We will follow them if they change jobs or change health care systems. We can follow them. It'll be harder to get their medical records. We can easily follow whether they're alive or not, because if they give us their social security number when somebody dies, it shows up in the National Death Index. We can also find out if they get cancer later in life through the state cancer registries. So it depends on what we're trying to do. Sometimes it's just general population. But all of this is the reason that these kind of cohort studies are way more expensive in the US than almost anywhere else. Because if you go to the UK, they have National Health Service, everybody's medical records are in the same place. And so it's a lot easier to follow. So some of these studies are more easily done in other settings. Yeah. We also do a lot of work with the military because of the military. If you have people who are career military, we usually can easily follow them for the rest of their career, even when they leave, because they keep getting in their health care from the military. So the military is a good partner for us as well.
We do have time for, I finally have the microphone. We do have time for another question if anyone has one. If not, let us thank Dr. Abnet again. And I thank you. I just want to remind people that, again, tonight, 7:30 p.m. For the general talk here with Dr. Abnet. So, thank you very much.
There's anybody here from Chem 104 or Chem 106?